certification issues, who should I have in the study, who benefits, who doesn't. All of these things aren't known, and you have to make some assumptions and some choices in the design phase. What would be nice is a lot of times we learn about this during the trial. And typically we only react to that knowledge when we're in between trials. So if you think about the phases of development, preclinical to phase 1 to phase 2 to phase 3, what happens is we run the trial. We're not saying that we do nothing while we're running a trial. Lots of stuff happens, but we don't change anything. It's nice and fixed. We stop, and then we look at the data and we evaluate everything. And then we run another trial, and we keep that fixed for a while, and then we look at the data and we change things. You can kind of think of this as a version, if you're driving to work, this is almost the version of, I'm going to point the car in the right direction, I'm going to close my eyes, I'm going to hit the gas, I'm going to go about as far as I think I ought to go, and then I'm going to open up my eyes again, I'm going to reorient the car, I'm going to close my eyes, I'm going to drive a little bit farther, and I hope by the end of the day that I actually get to the right place. Which, actually, probably, if you have the right kind of route, you're probably in decent shape. Adaptive trial design is aimed at the fact that while the trial is ongoing, we often learn things where there are obvious adjustments to make. And so, if we realize during a trial that one of our doses is obviously bad, can we just get rid of it? So those kind of adaptations, you can think of this as keeping your eyes open the whole way. If the car starts to drift, we try to basically get it back online. The kind of things that you would learn in a trial, so I mentioned the most basic is this drug doesn't work. So a lot of times you just find out this idea didn't work out. You can also find out that certain doses are promising, certain populations are promising. You can find out it's good for some and not for others. Good things can happen. You can find out the treatment's working incredibly well. You can also, so we talked about populations, groups of subjects that don't benefit. Adaptive trials are aimed at making use of that information and actually changing the trial design. So we may drop doses mid-trial. We may add combinations of treatments. We can stop for futility or success. We can stop enrolling certain populations. So if we have a drug for a stroke, we may find out, if we wait more than 8 hours to give this drug, it's ineffective. So we're no longer going to enroll patients after 8 hours. Those kind of adaptations. You can seamlessly shift phases of development. This is more relevant probably to industry than to academia, but a lot of times you hear of trials where they find a dose in a trial, and then they seamlessly move into a confirmatory phase, intended to save time. So the different kinds of adaptations that we might be doing, so futility stopping, these are just terminology. Futility stopping would indicate that we stop the trial and we declare failure. The goal is to save resources, to allow the subjects to go to another trial, simply to be able to disseminate the result that this doesn't work. We can stop for success. That's rarer, but it can happen. Arm dropping or adding, so we can talk about removing doses from a study, removing treatments from a study. We could add them. So you may start in a trial, the trial begins as sort of a proof of concept study where we come in with a high dose. If we find out the high dose works, so early on, then we may add lower ones. So our first goal is, do we have an effect? And a later goal might be, can we dial the dose down a little bit in order to solve that problem, or to get by with a lower dose. There's something called adaptive randomization. The example will cover this. This is a softer form of arm dropping. It's aimed at looking at the accumulating results and changing the randomization ratios. So if you've got a dose-finding study, you've got 4 arms in it. Instead of always allocating 1 to 1 to 1 to 1, you look at the accumulating data, and you may put a few more subjects on the higher doses and a few less subjects on the lower doses. So instead of just dropping it, taking it down to 0, we try to do kind of a softer form of that. It also allows doses to come back. So if a drug, a lot of times what you'll see with adaptive randomization is one dose looks really good at the beginning, you allocate a few more patients to it, you basically are saying, look, you look good, but you better go prove yourself. I'm going to give you a lot of patients. If it fails, the other doses can come back. So these kind of trials, you can almost see them searching through the space. This is looking good, didn't pan out, I'll look elsewhere, and those kind of things happen throughout the trial. An enrichment trial is aimed at changing populations. So I use the example of time since a stroke, for example. So we may be looking at what population, what is the sweet spot for the time, and certain populations we may omit as we go through. So we're enriching for a particular subset of the population. So the basic idea is that modern trials have lots of questions. As you answer your questions, your goal is to focus your resources on the things that you don't know. So you didn't know, a lot of things you didn't know at the beginning. You did what you could. What you can learn in the trial, make use of that. So if halfway through the trial, you would have gone, you know, I would have designed a completely different study. You try to pre-specify that. You go, look, if I find out this mid-trial, I'm going to write down before the trial even starts that I'm going to make these changes. I'm going to say, here's how I'm going to pre-specify my adaptations. So my first example is an Ebola trial that was designed for the Gates Foundation with DCRI Duke. Unfortunately, this did not get run as soon as it was ready. Ebola is still going on. There are about 30 cases a week, but there are no experiments on it at the moment. I will give a side story here. I was in the Toronto airport going through customs, and they ask you Ebola questions, and then they ask you, why are you here? What are you talking about? And I'm like, don't say Ebola. So anyway, so there are many different treatments. So no one really knows fully what to do with Ebola, but everybody has an idea about what to do with Ebola. So there was a lot of discussion about, we have multiple treatments. How do we account for that? We have a limited amount of subjects. This wasn't that rare a disease at the time, but it's still a rare disease. How do we look at lots of treatments? For simplicity, I'm just going to assume there were 4. For reference, there were more than 4, and they came in different domains, and this was a little more complicated. But I'm just going to call them A, B, C, and D. We're also going to allow combinations. So there was some thought of certain kinds of supportive care combined with antivirals might be good, and so the notion of having 2 treatments was on the table. So what we really have here is, if you think of A, B, C, D, and think of all the different ways we can combine them, we can do A and B, A and C, A and D. We can do A alone. That was one of the possibilities. B alone, B and C, B and D, C and... Anyway, there are 10. If I go through them all. I can do math when I'm sitting down, but I cannot apparently do it while I'm standing up. So the question is, well, what do we do about this? We've got 10 different possibilities. And I haven't put this up yet, but we really only have a couple hundred subjects. About 250 was what was the plan. So what do we do? One option is we can test these one at a time. So we could go, let's run a trial, small trial in A. If it works, woohoo, we got lucky, we picked the right one first. And then we'll see if B is better, but at least we found something. If A doesn't work, we'll go to B. If B doesn't work, we'll go to C. If C doesn't work, we'll go to D. And it actually became an interesting discussion of what order do you take these in. So if we find that B works, and then we find that D works, if we find B works and we find C works, do we go to D next? Do we try the combination of B plus C? You can imagine, it's a complicated endeavor, and you have to be pretty sure that you can order things well. So if people came in and they said A is likely to work and B, C, and D are unlikely to work, that might be a reasonable strategy. But here, lots of possibilities were plausible. The other thing you can do is, we'll call it the kitchen sink approach. So you could go through and have all 10 arms in your study. we had 250 patients. That only leaves 25 patients per arm. So that's not a lot. We got a mortality endpoint. 25 patients per arm, you can learn a bit, but it's on the smallest kind of sample size for this study. So the question was, is there any way to bridge this gap? So can we try to start with all of the treatments, all the 10 arms in the study, and focus our effort as we go forward? So we did this, and this is again a design, this did not get run, now sitting on a shelf somewhere. We adaptively randomized. So we start with some subjects on all of the treatments. So there are a few patients on all 10 arms. We actually only do 3. After that, we look at the death rates. And the doses, the arms, the treatment combinations that are doing well, we allocate a few more patients to those, and the ones that are doing poorly, we allocate a few less. Now, I haven't gone through the mathematical formula for this. It's important. So if you've heard of a trial design called Play the Winner, there's a famous trial, something called an ECMO, that did a very aggressive form of this. If you're too aggressive, you're worried about the case, an otherwise good treatment, the first couple patients die on it by bad luck, and you get away from it completely. That's the kind of mistake that you can make in this design. So you want to make sure that you do not adaptively randomize too aggressively here. But there's been a lot of work in this area on how aggressive is properly aggressive. This trial also had the ability to add treatments as we went along. So one thing that was important here is ZMAP, for example, if you remember that, there were manufacturing concerns. So we wanted a trial we could add things in as they became available. What we're going to focus on in this trial is the mortality rate in the study. So note, this is in the study. So we're talking about a disease outbreak. Treating the people in the trial is valuable. A lot of the people who are going to get Ebola potentially might be in this trial. And of course, we also want to talk about standard statistical things like power. We want to know the odds that we pick the right dose. So as I said, we're going to burn in 3 subjects on all the combinations. We're going to fit a linear model across all of the arms. So essentially, we're going to have a drug A effect, a drug B effect, a drug C effect, a drug D effect, and we're going to account for the crosses with interaction terms. Not doing too much on the modeling, but if you're into that, that's what we're doing. So let's see. I'm going to get away from the mic. I taught 14 years in lecture halls, so hopefully, can everybody still hear me? Okay. So this has a lot of stuff on it. I want to kind of hit the key points. The blue here, this is the truth. The axes are the different treatments. So if you see, here's A. If we combine A with A, that's a single arm treatment. So all they're getting is A. What we're saying is, suppose that in truth, that cured 40% of the people died. These are death rates. If we give A with B, then we also have that 40% of the people die. If we give, so here's B with C, 40% of the people die. So all of these are about the same. And suppose over here, if we give drug D, suppose that in truth, basically D does something and nothing else matters. That's what this scenario is. So D lowers the death rate to 20%. So that's the underlying truth. We're going to come in, we're going to adaptively randomize. We see 30 patients in all the arms. The ones where people live, we give a few more. The ones where people die, we give less. What you're seeing for the dots, this is the average number of people who are in the arm. So if we think about running this trial over and over and over again, what it's going to do is it's going to move towards the arms that are doing well. Now, in any individual trial, it can get the wrong answer. These are averages. This is what power and type 1 error give you. So on average, what happens is every one of these arms in D, you're seeing we're averaging about 50 patients each. This one's a little bit lower. I think this is just simulation error. But anyway, we get almost 200 of our patients get D in some form. At the beginning, they're spread all over the place. But what happens is early on, we've got 40% of the people are dying in these combination arms. So we get away from them. We don't assign patients there. The dots are colored to give you an idea of the death rate. So green is live, red is die. And so you can see these arms, lots of green. These arms have more red. So purpose of the adaptation, get more people to where the good treatments are. If you look at for treating the patient in the trial, if we just did a fixed trial, we put 25 per arm in all 10, we have a lot more patients out here. So instead of getting 8 and 11, we end up with 25 each. If you do a fixed trial, on average, 80 people die in this study. If you do the adaptive trial, on average, about 61, 62 die into the study. So that's 20 extra lives saved just from the statistical design. The other piece of this is what are the odds that we choose an arm? And so this number at the bottom, this is the probability that we say that this arm is the best. So you can see about a 28% chance we choose this as the best here, 28 here, 29 here, and 13. So there's actually, the model kind of has a predisposition to choosing combinations. Any of those is the right answer. So by and large, we will say that dose D is good. Now we're not really clear on what the combination is. That was viewed as an acceptable weakness of the design. If we continue further, we could try to figure out any preference toward a monotherapy as opposed to a combination. Let me give you another scenario here. So by the way, Lori, how long am I actually going to talk? I know we started a little late. I can make this as long as you want.
You can go for, I mean, you can keep going. I guess so I'm thinking it's about 2:15 right now. So you have until 3:30 officially, but we started late. So
Okay, I will try not to go too much because I know
like
people want.
3:40 is good.
All right, I will do that. Let's see. So here's another scenario. This is a situation where if you look at the truth again, drug A is harmful. Drug A is actually killing people. There's a 50% death rate. Drug D continues to have some advantages, but there's a sweet spot here. If we give D and C together, then only 10% die. So we use the same design, but now what happens is you can see we really get away from drug A early. Patients come in, we see a lot of deaths, we don't randomize to drug A anymore. Other arms, some of them here is a 30% death rate. We explore these for a little while. You can see here's 27, here's 26 subjects on average. But what happens, actually over half of the study tends to be in that sweet spot. Because the death rate is so low, the trial, you don't wait for 250 patients to come through here. This arm is doing really, really well. Let's put all the patients there. And so you can see lots and lots and lots of dots. Very few of them are red. And here, we end up saving about 40 patients. In addition, if you look at the power of the trial, this trial, if you do the adaptive design, it identifies this as the correct arm 99% of the time. Has 99% power to actually figure out that that's the right answer. The fixed trial, if you just put 25 per arm, it does a good job too, but it's at 91. So you can miss this in the fixed design 9% of the time. You miss it 1% of the time with the adaptive trial. So one of the purposes of the adaptive trial is I'm able to put more resources on the doses that are doing well, the combinations that are doing well. I get more power for discerning that best arm. All right, lots of scenarios. The slides should be available. We didn't just simulate this under a couple different scenarios. We simulated it under many. trying to make sure that the design worked no matter what the underlying truth was. All right, so let's see. So I need to switch to that's an example of an adaptive trial. I'm going to switch to trial simulation. So I'm going to call this a complicated question. One difficulty here is I want to be able to do the math so I can verify things are right. But of course, then I have to call things complicated that are not quite necessarily realistic. So I'm going to start with this quote, complicated question. If you flip a fair coin, it could be 10 times. Anybody know the probability of getting a streak of 4 heads in a row? I actually figured you would know. So I had no idea. So anyway. Anyway, it's actually 24%. If you took a class on Markov chains, you could solve this thing. I had to actually play around with it to get it. So the 24.5% is actually pretty likely. If you ever ask somebody to generate random numbers and just write them down, one of the things they almost always do wrong is the streaks aren't long enough. You get lots of streaks at random hands. So anyway, 24.5%. So that's if you know the math tricks in order to get it. If you have a handy computer, there's a simpler way to do this. Takes a little longer, depending on your definition of longer. But let the computer run the experiment lots of times. So we just tell the computer, flip a coin 10 times. They know how to do that. So I did simulation 1. I got tails, heads, heads. Anyway, I didn't get a streak of 4 heads. So that's a no. The second simulation, I got this. Oh, there's my streak of 4 heads. That's a yes. Here's one, I got a long streak of tails, but I didn't get a streak of heads, so this was a no. And I just did this over and over again. 4th time, 5th time, 6th time. And I kept going. So there's something in statistics called the law of large numbers, which says if you simulate something over and over and over again, you eventually get to the right answer. This is why casinos in Vegas can always beat you, because they do enough bets. This is why insurance companies can make a profit, except when a hurricane comes, because they make enough bets, and so on. So I ended up running 100,000 simulations of this. After 1,000, the rate was 24.2, 24.7 after 10,000. After 100, it actually converged to the 24.5 and had converged actually a couple more decimal places down. So this is one mechanism. If you want to find out a complicated probability, this is a way to do it. Get the computer to simulate what happens and just count up how many times does this event occur. You can also do questions. I don't know the math on how to do this one. So if I were to flip the coin n times and look for a 4-head streak, how many times do I have to flip the coin that gives me a 90% chance of such a streak? This is actually really similar to the question you get, tell me the sample size you need for 90% power. The same kind of calculation happens. If you're doing a standard, whatever that means, statistical design, there are equations for this. If you do an adaptive design and you say, what is the sample size for 90% power? I don't know. What I have to do is I have to go through an exercise like I'm about to do on the next slide, which is just, I'm going to simulate different n's and figure out the value. Anybody have a guess? 20. Okay. It's 65. For what it's worth, I wouldn't have guessed that. So, anyway, so all I did is just by simulation, I got n equals 10 is 24. I tried 50, I tried 70, I tried 65, and it just happened to work. So if I had had to, I would have kept searching and found the 90%. All right, so let's go back to reality. Some semi-reality anyway. So in what I have in quotes as simple, and I don't necessarily mean simple, but in simple clinical trial designs, we can often do the math directly. this is an interesting question of what you mean by simple. I once had a professor once who said it is trivial to show, paused, stopped for a long time, went back, looked in a book, stood there, and then went, yes, it is trivial to show. So anyway, it's always your definition of what trivial is. But anyway, you can go through the math directly to get power and sample size if you've at least done it before. There are ways to do this. In complex trials, and this isn't, there are lots of non-adaptive trials that are complex, but in most adaptive trials, we have to do simulation to get things. If you ask, what is the power of this trial, I have no equation I can put things in. I have to get the computer to simulate it to get the answer. So here's an example of this. So let me start with a trial with 2 doses. So a low and a high. I'm also going to have a placebo arm, so I mean 2 active doses. I'm going to enroll 216 patients in total. I'm going to enroll 36 patients per month. I'm simplifying this with deterministic accrual. People who do this realistically. So they assume ramp-ups in accrual and assume the endpoint may be delayed. You can do this in a lot of generality. I'm keeping this basic. So I'm going to assume that you have an instant endpoint. So as soon as the patient comes in, might be, if you're talking about a migraine study, it's about the closest thing in medicine you get to an instant endpoint. 2-hour pain relief is one of the endpoints. The endpoint is a composite event. So this is a situation where the low dose can actually be better than the high one. So there's a situation, the thing modifies it, but something can go wrong. The low dose might be better, the high dose might be better. I also have a scale. This is actually a real trial, but I've censored a lot of the details. If the increases of 2 units are considered valuable. So medically it's important if we get to 2. So here's a basic trial design. I have 216 subjects. I got 3 arms, control, low, high. Let me just put 72 patients per dose. I'm going to have to put an alpha adjustment. Michelle talked about multiplicities, so this is a Bonferroni adjustment. I'm using alpha over 2. There are slightly better ways to do this. This trial always enrolls 216 subjects. Suppose the drug doesn't work. So what's going to happen? This is where the true effect in low is 0. So the difference from low to placebo is 0. The difference from high to placebo is also 0. So no effect. The probability of getting a successful trial here, I can do the math, this is about 2.3%. That's the type 1 error rate. And this trial always goes to the end. I always enroll 216 subjects. And so what happens is essentially all the time I get the whole trial enrolled and almost all the time I fail. So that's a bad kind of arrangement. On the plus side, kind of, if the drug does work, what I've got is my true effect in low. Let's just hypothesize. Suppose it's 1 in the low arm. That's not great. Remember, we want to get to 2. And suppose the true effect in the high arm is 3. So the high dose actually is good. If I run this trial, I have a probability of success of 77.1. So that power is not 80%. One possibility, if you're interacting with your friendly local statistician, is to go, okay, if 72 per dose only got me 77% power, can I just do like 76 per dose? I've got 80% power, and then I can go write my grant number. So, which is a perfectly plausible thing to do. So because I'm up here talking about adaptive clinical trial design, you can guess what's next. So I'm going to call it a naive adaptive trial. I'm not trying to pull out every bell and whistle, but I'm going to put in some interim analyses. So after 1 month, I'm going to stop the trial if neither dose achieves a 2-unit increase. And after 3 months, what I'm going to do is I'm going to choose the dose with the higher observed beam. And then I'm going to continue enrolling the other one. So here's what I'm trying to do with these. I'm trying to address the fact that if my drug doesn't work, I want to get out. So if it doesn't work, I'm giving myself a way to get out of the trial early. The other thing I'm trying to do with this is to get at this notion of increasing power. If after 3 months I have an idea which dose is better, if I can get rid of the other one, I can allocate more subjects on the dose that's left over. That increases my sample size on that dose and potentially gets me more power. I don't know if it does yet or not, but that's the hope. And at the end, I'm going to do the same analysis I did before. I'm going to run a t-test, and I'm going to take my Bonferroni penalty, and this may be a little extra conservative, but I'm not going to get into that. So example, if I come in, so suppose I come in, month 1 data, you can see that this is 4 higher, this is 3 higher, this is great. I want to get 2, I'm doing better than that, so I continue. At month 3, I do a dose selection. So I come in, the low dose is doing better than the high dose. So I choose the low dose to continue. And then at the final analysis, I've gotten rid of the high, but I compare my low dose results to my placebo. And it turns out this would be a successful trial. The p is 0.001. I haven't written it down. The standard deviation is about 6. Here's another trial. If I come in, I've got a placebo of 6.8. The low dose has no observed effect. The high dose only has an incremental small effect. This doesn't hit my futility bound. This isn't good enough, so I just quit here. That would be a failed trial, but it would be a failed trial after 1 month instead of a failed trial after 6 months. So how does this work? This actually works a little bit poorly, and here's why. So I'm going to simulate this trial. Remember back to the coin flips? I basically flip 10 coins, look for a streak. Flip 10 coins, look for a streak. Did it over and over and over again. I'm going to do the same thing here. I'm going to simulate 1,000 different trials. I'm going to simulate to month 1, figure out whether they were futile. I'm going to simulate to month 3, figure out what dose I select, and then simulate out to the end and run my t-test, and I'm going to count how many were successful. So the first part of this, let's look at the futility. I want the trial to declare futility if the drug doesn't work. So let's assume no effect, and I'm going to simulate 1,000 trials. So there's 1 dot for every trial. So 1,000 dots on this. Here, for example, is a trial that the low dose, this is the observed low dose effect, it was about 6. Now, the y-axis is the observed high dose effect. It was also about 6. This would look like a great result early. So this is not futile. In contrast, the results over here, remember what has to happen for me to be futile. I need the low dose to be below 2, and I need the high dose to be below 2. So any trial that's in that red region, neither dose looks good, I'm going to declare it futile. All the trials out here, I'm going to keep going. So this isn't bad. This actually declares futility 679 of 1,000 times. So if you ran this trial about 68% of the time, if the drug doesn't work, you get out in a month. You save all that time and effort, the subjects aren't exposed to anything. That's a good result. The difficulty is that you find out that suppose the drug does work. So let's get back to this situation. The low effect is 1, the high effect is 3. Well, now what happens? So now we end up in a situation where we actually stop 305 of the 1,000 trials. What you're seeing here is look at the amount of variation. Even though the drug is truly effective, so the high dose has an effect of 3. So the high dose, that's this axis. The truth is this line, but in actuality, you can get values as low as -3, -4. You can get values that are up to 10. By random chance, a lot of trials get stopped for this. All of those trials might have otherwise been successful. So you're stopping things incredibly rapidly. Those 30% of trials That's an immediate hit to power. Only 70% go on. Not all of those are going to be successful. So your power can't be any more than 70, and it's probably a lot less. You can see this another way. This is a simulation version of the graph Sharon was showing on Monday. So I've tried to put, there are 2,000 dots on here. The dots in purple, these are the simulated trials that we saw when the drug doesn't work. The green dots are what you would see if the drug does work. So the main point I want to make here is there is a lot of overlap between those two clouds. So if you just saw a dot, you don't know, does it go with the green ones where the drug works? Does it go with the purple ones where it doesn't? If you're going to put in a futility rule here, it needs to be a lot lower. This was at 2. It needs to be a lot lower down in here, so I'm only getting rid of the purple dots where the drug doesn't work. I don't want to be screening out good drugs. I only want to be screening out bad ones. So I'm going to put in a new futility rule. So I'm going to I want to avoid stopping drugs. One way that I'm going to do that is I'm going to look later. The fact that those two clouds overlap so much really just means 1 month is too early to do this. I can't discriminate good from bad at 1 month, so I'm not going to. And that's what the simulation's showing you. It's just too hard at that point. So I'm going to move it to month 3, and I'm going to do it with the dose selection. I'm also going to change the form of the rule to something a little bit more statistical. And I'm going to, if neither dose has a p-value less than 0.25, then I'm going to go ahead and declare futility, rather than just saying 2. So what does this do? Well, now, this is what those clouds of points look like at 3 months. Key point, 1 month they hadn't separated. At 3 months, they're starting to separate a little bit. So here's the kind of results you get when the drug doesn't work. The purple dots, they're all down here. When the drug does work, you get the green dots that are up there. It's not perfect, but now you can see the separation between them. It makes a lot more sense to put a futility rule. That rule's about right here. A lot of the purple ones are excluded, and I don't mistakenly get a lot of the green. So I'm really trying to do that discrimination. All right, do I say, yeah, so I have to be careful with the main point. So the nice thing here, only 5.6%, 56 of those green dots are killed here. So you can make a mistake, but it's not 30% of the time, it's 5. And we still declare this trial futile 62.6% of the time. A lot of purple dots are killed early. All right, almost done. Collect my thoughts, figure out my 3 minutes. All right, so let's see. Let me focus a little bit more on the good scenario. So suppose the drug works. So this is the low dose has an effect of 1. The high dose has an effect of 3. That's what I mean by truth 1, 3. At 3 months, remember I had that futility rule. So the futility rule only kills these trials now. I got my separation. So these are mistakes. These are situations where the drug just randomly was bad early. So 56 of these 1,000 dots are futile. 944 of them continue. What was the second part of my design? I was going to pick a dose. So if I pick a dose, I'm picking the better dose. So this is the line x equals y. If I'm above the line, I pick the high dose. If I'm below the line, I pick the low one. So again, I do a pretty good job. 871 times I pick the correct dose. And there are 73 times where I don't. You can't do dose selection perfectly. Apologies. But so if I get down into the simulations, and so yeah, I'm in good shape. All right, so I'm going to just tally. So I've talked about the futility rule. I've shown the dots for that. I've got a line which lets me see how the dose selection happened. I can also simulate to the end and see what was successful. So you can see for ineffective drugs, I screen out 626 early. I pick the low and high kind of randomly. Only 20 of these trials are mistakes. You don't want to declare a success when the drug doesn't work, but it can happen 2% of the time. In contrast, if the drug really does work, I only screen out 56. I primarily pick the high dose, and the high dose is typically successful. So I'm successful 820 of those dots, 82% of the time. So to conclude, a comparison here, if you think about where we were and where we are now, I had a fixed trial that had a 2.3% type 1 error rate. It's actually lower now. It's 2. I haven't actually proven it's lower. I know Chris is looking at me calculating sample sizes, and it's absolutely right, but that's not a proof yet on 1,000 cells, but it is lower. The power has gone up. So 77.1 for the fixed trial, I'm now at 82. Part of the reason is actually this bottom line. On the selected dose, I used to have 72 patients. Now I've got 90 because I dropped one. I was able to, at the end of the trial, I was enrolling 2 doses, not 3. And finally, if the drug doesn't work, the fixed trial had no savings at all, and the adaptive trial saved half the study 63% of the time. So, and you can mix and match these things. So an adaptive trial, you can take the features you want, don't take the features you don't. If you don't want to do dose selection, don't do it. You can still have a futility rule. So decide what's best for you in that time. No, I'm running out of time, so I'll just end there.
Thank you, Kurt. So we have questions?
Are you allowed to ask a question?
I would be delighted for Chris.
Well, this is not so much a question as I'm wondering whether you'll talk about something I think that comes up a lot, and this is really your first time, the adaptive randomization. And like academic trials, one of the things that we found is there's a lot of variability across IRBs at academic medical centers when it gets to like the informed consent. And some of them want you to tell like the prospective patient your probability of being assigned drug, placebo is X percentage, and some of them are more flexible. And so with adaptive randomization, sometimes you end up kind of struggling with how to make that wording work with the trial because you don't know for a given patient in the study what their probability will be. So I was curious if you could maybe address that, because I could see that coming up if any of these students are using it.
Yeah, so I can't say a ton about it. I mean, you're certainly right. You get it both ways. I don't have as much experience on the academic side. For the industry trials, we've usually been okay in earlier phase trials where there's an argument. We're learning. The usual counter-argument that's made, there was a JAMA editorial on this, is that by doing the adaptive randomization, we're treating the patient in the study better. That often carries a lot of weight. There was an interesting, there's an article by Hay and Kimmelman on the ethics of adaptive randomization. They focus on 2 arms, where I think it's actually more controversial. I think you really get benefits for 4, and that's not in the article. But I think this will remain somewhat controversial. So I don't have an answer for you, but it's definitely an issue.
So Chris, I can tell you what we're doing in SHINE. We're doing it in SHINE. The consent say at the beginning of the trial, you will be randomized 1-to-1, and then at some point, you'll have a slightly higher likelihood of being randomized to whichever group is doing better. And for the most part, I think most of our IRBs didn't have a problem with that. It may be that some of the IRBs required a little more specific about what is slightly higher. And what we tell the patients is that we don't know which treatment is better, but you have a slightly higher likelihood of getting the better treatment than you do of getting the less good treatment.
The advantage to this is you never want to be the last patient enrolled in a clinical trial. You'd rather be the first patient afterwards, because the last patient, if you've done it fixed, you're still on that 1-to-1 randomization. The next person gets whatever one. So the adaptive randomization has the advantage of, if you're at the end of the trial, you get the benefit of all the information.
Although you could turn that around though and say you don't want to be the first person in the trial.
No, that's absolutely true.
Right?
That's absolutely true.
Wait to have your strip.
I have more a practical question. We tried that in oncology, in phase 2 trial and multi-center design, and there really became an issue about reporting, because obviously you make your decision based on the events, and if there's no quick reporting from the centers and the data is not very good, it can be an issue. And another point is like the trial we did took probably 5 times as long as it would have been traditionally, because you always had to wait until the events happened. So if it was just traditionally, there would have been enough centers who wanted to enroll patients, but because we always had to wait until we had enough events happening to adapt the design, it took much longer.
So I guess that seems unusual in my experience. So you do have to spend a lot of time on logistics. If you're not prepared to run these, you can have disasters. Now we've run a trial. So last year we ran a trial that had 78 interim analyses. It did adaptive randomization every week. And it was set up so Sunday night, a computer queried the database. It generated a report. On Monday morning, that report went to a board, which had to look at it and approve. And by noon on Monday, the adaptive randomization probabilities, they were adjusted, and it went back in. Now, that took a lot of effort to get there. So I guess I certainly agree this can happen. And you want to talk to everybody involved, your CROs, every one of the sites, and you need to make it clear, we're going to need data entered faster. We need to do this continuously. We can't wait, the interim analysis is coming up, let's now pull out all the CRFs and type them in. So that won't work. So you definitely have to make sure that everybody, all your stakeholders are logistically on board before you do this. Yeah, so that's the other thing. Keep in mind, adaptive designs never look at the entire data. You don't need to clean the pregnancy test to do an adaptive design. You need to get into events. Yeah.
Yes, so question, are adaptive designs appropriate for patient groups that are fairly heterogeneous? So say, you have some sort of patient characteristic that predicts response to your treatment. Is there some sort of adaptive design that's actually stratified or takes into account these sort of predictors in terms of saying is this a response to the treatment, if it's not like mortality or something?
You could absolutely put in covariates into the analysis that would represent the strata. In the Ebola trial, we didn't do it, but there was actually a lot of discussion of adults versus kids, whether we wanted to do something like that. If you were working on a trial where there was a genetic marker and there were multiple sets, you can imagine a situation. So there's a big trial called I-SPY 2, which is in breast cancer. And it actually does adaptive randomization within genetic types of cancer. So HER2 positive cancers are randomized differently than people who have a different genetic signature. So you can absolutely do that. It's more complicated, but yes.
I was wondering if you could comment about the role of adaptive clinical trial approaches to diseases where your endpoints might take longer. So, let's say neurodegenerative diseases where maybe it's going to take 6 to 12 months or more maybe to see an effect of a specific drug. Can you even use this approach in this population?
So the time to endpoint is important. If you're going to accrue your entire trial early and then you have to follow for a number of years, and you aren't going to know anything, you really can't do an adaptive trial. So the key quantity is always the ratio of the accrual rate to the endpoint time. And if that can go against you. In a lot of trials, if you have a biomarker, you would often adapt on the biomarker. So this trial was actually, the one this came from, this was a biomarker trial. They were picking a dose and then they were going to follow patients for a long term. I didn't have that expertise. The I-SPY trial, for example, that I was talking about, it is aimed at, it has measures of tumor change, MRI images, and they try to predict the longer term endpoint. So it's good to have the endpoint. If you can get a biomarker that predicts the endpoint, you can do some. And it's just a matter of how predictive.
Can I add something to that I think is
Yeah.
important? you want, I mean, the time to measure the endpoint is important, but the other piece that gets lost in the conversation a lot is you have to have high-quality data management that gets that information clean, queried, and ready to go. Because if that slips through, it doesn't matter whether you get the endpoint quick or not, right?
Yeah.
Great, thanks.